Well, welcome everybody. I'm happy that I could present something here. So a lot of the work here is actually not my work, but also with Detmer, but also some other people here. And similar to yesterday, I think I will raise more questions than answers. And like what Serena showed now, I think we know quite a lot about the profit transfer and the composition of kit rates. I think this is a topic that we actually really know very, very little. So I hope we will have some more discussion and questions instead of me showing proof, because simply there's not so much to show maybe. So, oh yeah, what I also want to say is we had with Kitworks, we had some workshops before. I think Michael has been there, Silke has been there, Detmer, Elena, Serena, and I'm very happy that we have this workshop because I think it's a very, very good way to share ideas and to get inspired to do new stuff. Because if I think at all the publications we have with Kitworks, I think many of those ideas or collaboration started at workshops. So take your chance. Yeah. Another workshop. So some of the work that we will show today with Detmer, we did at the C-Sync. So it's probably something that will be cut now in the US, I guess. But they fund new science. So we try to integrate modeling with experimental work. Then some other people also here. You may know some of them, some students. These people also part of the workshop. And Silke is also a little bit in the end in an experiment from that Detmer did with the students. So yeah, Elena was my co-promoter, Detmer was my promoter, so did my PhD here at NEO. Then I went abroad a bit and then came back for another postdoc. But right now I'm a teacher, basically, so I'm here for fun, as I said yesterday. So
yeah, it's nice. I didn't mention it in the announcement, but thanks for being here in your holiday.
Oh no, that's not the point. I'm happy that this was in the holiday because otherwise it probably could not come. So good planning. Okay, so as I was the last one in the riddle of the talks yesterday and today, I want to also recap a little bit because I think many of the topics also will come together, and I think we will just start with the basics. I know this is not a plant, but I just put a picture there to make you think about what does this plant or microbe need to grow. So think about what we did this morning already, but also yesterday. Nutrients. Which nutrients? Okay, phosphorus, nitrogen, carbon,
iron, vitamins.
Vitamins. Okay, getting closer. Light. So that's maybe light to carbon, but yeah, they needed to make the carbon. Any other big things?
Water.
Water. Honestly, I just made a quick list of what came up in my mind first. So I also had water, nitrogen, phosphorus, oxygen, silicon, of course, silicate is maybe important for the diatoms to make the shells, the trace elements. Some of the with Kitworks I actually don't know, but I suspect that some of them may also not grow without trace elements and vitamins, light, etc., etc. So as you can see, there are so many things that we did not study yet. We only focus on a very small thing. One other bridge I want to make is, so you have these resources and nutrients or environmental conditions. What does the plant need them for? What do the algae need them for? Which functions in the cell?
Photosynthesis.
Yeah, so photosynthesis. Why do we need photosynthesis?
Carbon fixation.
Yeah, so carbon fixation and growth, probably, right? Any other? Maybe that's already the biggest one, growth. Any other functions? Specifically, like nitrogen and phosphorus, what could we use it for more specifically? Or a plant? Exactly. So, which is also part of growth, but that's indeed already more specific for nucleic acids. And when we think about fungi, chytrids, what do you think they might also need them for? Nitrogen and phosphorus?
Reproduction.
Mm-hmm. So what will they use it for? Nitrogen?
Protein building.
Yes, or the what else? Like that Serena just showed. Sorry? Mhm. But of course, they also need enzymes then to digest hosts, right? To actually consume them. So that's another function that the nitrogen can't have, or phosphorus for the replication. So very short on the nutrients and their functions. Well, that brings me to Liebig's law of the minimum. Maybe familiar to some people. Does anybody know what it? Does anyone remember what it means? It's pretty simple. Antoine, I see you shaking. That's why I told him before I would let him do the talk. So.
Yes, how we say, the growth is always limited by the factor that is less the scarcest.
Yes. So that's why this barrel is nice. So when it's the overflows, of course, it overflows at the point that's the scarcest, right? So that's also what limits then the growth. But obviously, this is pretty much a simplification. So again, thinking about this morning and yesterday, do you think this is relevant, Liebig's law, to our host-parasite interaction?
Should be.
Should be. Could be. Could be. I think indeed it could be, but if it does, I actually wonder, because obviously there's a lot of other things that could limit the cell or dictate whether the nutrient becomes limiting or not. And obviously, like what I said previously, you will be able then to show that in the growth or in the composition of the cell. But first, I want to let you think a bit about which factors could complicate. So if you want to translate it to nature, which could complicate using Liebig's law, and I want you to discuss a little bit with your neighbor, like in two minutes or so. I want to do a quick round. What do you think? Why we would be able to use it or not use it?
Bioavailability. Do you have the enzymes to do it?
Take and stuff like that.
Bioavailability enzyme. You might need a lot of energy to be able to process it, and you don't have the energy.
The total growth is only
Like with the fatty acids, you can't synthesize a bit like with amino acids as well, where you can't synthesize every single one. That's not to do with the elements available, is it? It's to do with more exactly, yeah, whether you can or can't make it. I guess then you could say that unit is still a level there, I guess.
The slowest in the team because it's going to be the slowest
Competition for sort of implied.
Maybe we can do a recap. I heard some good things already. Do you want to maybe we do a round and then say something somewhere? I guess we will overlap quite a bit. So what's the most common one?
Bioavailability, sort of whether you can actually extract the element that is there.
So that it's present, but you cannot.
Exactly.
No uptake. Okay, yeah.
Variation over time and availability of different nutrients.
So that was yesterday. Who said that again? I remember, but in one of the talks also that the environment keeps changing continuously, right? So it's hard to study that. Serena and.
We are considering also the multifactoriality. There can be more elements that could be limiting that can happen.
Yeah. That also matches with what Joe said. So and then maybe antagonism, right? That one nutrient more inhibits actually uptake of another, or the other way around. So there may be interactions, yes. Somewhere there in the back.
Yeah, we also discussed the viability and the ability to extract the environment.
Mm-hmm.
Also thinking for limitation.
Alternative metabolic pathways to use.
we were also discussing light limitation. Yeah.
So the changing over time, or the yes, yeah. I think honestly, I also didn't think much about this. I just put some ideas myself because I don't think you can make this list complete, right? It would take a very long time. Time and space. So I think we heard already, right? I mean, time changes continuously, right? So space is the same. The algae move to the water, or move to different habitat, or the wind or the flow brings them somewhere else. So I mean, Liebig's law kind of assumes that it's stable, right? Which is almost never. Well, there's a lot of non-nutrient factors. We are talking about nutrients, but of course, other things can affect uptake of nutrients or the bioavailability. I think that matches nicely. Another one is the interacting. We also heard already, so it can be co-limitations or the synergy that they uptake together or opposite. One other one that I think is very interesting is the plasticity. So that's obviously to counter all these things. So some cells can store nutrients or buffer them, or maybe even collaborate. I don't know, but if you multicellular, I guess that's possible. Or in a filament, or with some algae that have nitrogen fixing or whatever, they could. Some of them may help actually against this. And then we have the relative availability. So besides the interaction, the relative availability may also matter. So that some nutrients need to be in the same concentration range, whatever, to, I don't know. I'm not going to talk about any of them. I'm only going to talk a little bit about these last two. But keep in mind, all the things that we did are under stable conditions and in lab conditions and field cultures, and but not much there. So if you are looking for a new project, I guess there's still a lot to do here. Well, that brings me to stoichiometry. So that's, yeah, Detmer obviously works a lot on this. So that fitted nicely when I started here with the topic of host-parasite interaction. So the stoichiometry works is that you have a certain balance in elements, and obviously there is a chemical reaction or whatever happens, and it's like a mass balance. The elements, they don't disappear. So you can use this balance also in the algae or the food. I mean, everything has a certain ratio. One thing eats the other, and then something happens, and maybe there's still some waste. But you could also look at this as a mass balance. So the stoichiometry is this ratio, and ecological means that there is this interaction between the organisms. So yeah, this is also one of these key books from Stern and Elsar. They show a lot of examples on this with herbivore, but also just with algae. So how does it work in the food web? So what we generally see is that here you have different groups of organisms. So primary producers, grazers, secondary consumers, and then you have the CTP ratio. So the CTP ratio means the carbon to phosphorus ratio. So if there's phosphorus limitation, CTP go up or the opposite. So this variation may reflect also limitations in nutrients in the environment, and these algae there, they can handle it easily, or actually generally autotrophic organisms can handle it relatively easily, but also generally they are a little bit higher, the CTP. Now what we see then, if we go up the food web to the heterotrophic organisms, we see that generally they are a little bit lower, but mainly they're also a lot less flexible. So with the plasticity in the previous slides, that makes sense. They have a lot more structural compounds or other things limiting their body to be plastic. Well, and what you can also see from this is that sometimes there seems to be a mismatch between the food or the grazer. So obviously this brings questions. This may be energetically very costly to eat something that's very different from yourself. And therefore, coupling back all the way to the beginning of the talk again, to the functions, you may be able to measure that in functions like growth or photosynthesis or whatever. And what we obviously wondered, there was quite a bit of work on this already, how would this work for host-parasite interactions? So one thing we discussed earlier, we expect chytrids or fungi to be rich maybe in nitrogen and phosphorus, and the same for viruses, simply because they're small, they don't have much else than a lipid globule, right, and which is rich in nutrients. So we expect this ratio to be low because it contains maybe a lot of phosphorus. Well, and this is an example how that may look like. I mean, this is on a growth rate of grazers on algae, but you could expect that there's different ratios which the consumer will like or dislike. So in this case, the grazer likes this ratio because it can then grow faster. Maybe it's more similar to its own food, so higher growth rates, and the opposite true. So when there's very little phosphorus, the growth rates go down, and when there's maybe too much phosphorus, the growth rates also go down. You could compare it to yourself eating very unhealthy food, right? If there's too little or too much of something in your food, it will affect your growth too. At least that's the hypothesis. So each organism has an optimum in this ratio, but also in the functioning that is relates to that. Well, and then we, a few years ago, this is an output from this workshop that I showed at the C-Sync. So what we did, this is a hypothetical paper because we have very little data. We just try to start with theory. So what do we expect? So again, this is just a certain carbon to neutron ratio, and you can see here in the background again. So that is the plasticity, in this case, of a plant or algae. You can see it can maybe change their ratio very easily. But then just two examples, two different pathogens. You see they have different requirements, and they're also less plastic. And if you then compare them, this may lead to some certain mismatch between the pathogen and the host. And these mismatches may also differ between different parasites and their host. Well, then hypothetical scenario B, we will have nutrient limitation. So what will happen is this carbon to nutrient ratio will go up because less nutrients, so it goes up. Well, the algae or the adsorptive host can shift easily, as we just discussed. It will go up. However, these pathogens, they can only be plastic until a certain moment, and then they cannot change their stoichiometry anymore. And in this case, it may lead to larger mismatches with nutrient limitation. Well, the opposite may also happen if we add nutrients or if we lower the carbon to nutrient ratio. Again, the algae can handle easily, but in this case, in this scenario, maybe the pathogens could also benefit from that, which may lead to a small mismatch or no mismatch at all. And obviously, then we would expect that the pathogen will be very happy. So this brings us to the hypothesis. These pathogens may be more sensitive to nutrient limitation than their host, and we may see that then in the reduction of the infection or infection rates. Well, a lot of theory. So knowing all this, how would you now demonstrate this nutrient limitation in the host or the parasite? I mean, I just gave away already the growth rates, but besides the growth rates. So the elemental composition, yeah, that's a good one. Any other way to prove that something is nutrient limited? Bioassay. Bioassay? This goes too quickly. But correct answer. So I just put a random carbon nutrient analyzer here, but indeed, this brings us back to this picture in the beginning. If you measure the particulate amount of nutrients in each organism, but also in the water, you could make this mass balance and maybe show that something is nutrient limited. Because in the waste, if something is nutrient limited, probably in the waste, this element will also not be there or very low. And one of the other elements may be very high. So you could maybe show probably this one's more limiting than the other one. And obviously you can do bioassays, which means adding the nutrients or removing or diluting the culture. That something may become limiting. And of course, then the next step is growth. So the combination of these measurements and measuring growth throughout time. See here, this is a microbial example, but it's the same for cyanobacteria or plankton. You can see generally in the beginning, when you dilute them, maybe they're not happy yet. They have a little bit of stress, maybe even. That's called a lag phase. Then they start accelerating growth. Then you have the exponential growth, etc., etc. So in which phase would you expect that you would see the nutrient limitation here?
In the exponential phase.
In the exponential phase? And where in exponential phase? Because how can they grow exponentially if there's no nutrients?
At the beginning.
Yeah, so somewhere halfway, right? So it's like this inflection point, how you call it, like that it goes from the highest growth rate to the lowest growth rate. So what you will often see in the papers, and if you also write papers, what you have like this, we call it the maximum growth rate. And after this maximum growth rate, usually, please correct me if I'm wrong, Dagmar, but usually after that, some nutrients should become limiting. So this is also why we often do our experiments in this range, because over here stuff starts dying. So nutrients are maybe released again, but here there's so much nutrients that you can maybe not even show the limitation or study it properly. So this is a way that we try to handle this variation in time and space a little bit by controlling which growth phase we are looking at. Another one is obviously we will get a lot of rates, which, yeah, I know Andre was yesterday here, but also Serena, or others maybe online, but these rates make theoretical ecologists very happy because yeah, this is also one part that is still necessary, which would be a nice way to continue working is by getting just more rates of more organisms in more conditions, which we could then use to improve maybe predictions in modeling or whatever. Okay, more hypotheses. We already discussed, so the cell takes up nutrients. We focus on nitrogen and phosphorus. We assume that more nitrogen and phosphorus will stimulate growth of the host. Well, these nutrients are also used for parasite reproduction. So we have the chytrid viruses, so there may be some competition for a nutrient in a cell, I don't know. The parasite reproduction will negatively affect the host growth rate. Yeah, which parasite do you expect to be more sensitive to which nutrient? I think we discussed it a bit already, but do you expect, for instance, a difference between chytrids and viruses or fungi and viruses? That means no, everybody thinks...
You're asking for it.
Probably. A good guess. What would you guess? Maybe the viruses are more affected by nitrogen, because they have a lot of genetic material produced.
Mm-hmm. Yeah, could be. And the chytrids? I'm just going to wait.
I'll try again.
Yeah. So our expectation was actually that because of the enzymes and the mechanisms that chytrids need and the globule, we expected that they may need relatively more nitrogen than phosphorus. And for the viruses, we expect actually the opposite, but mainly because they have such extreme high growth rates. Can you call it growth? Yeah, maybe. But the growth rate of also like things that grow faster, etc. They usually require relatively more phosphorus. So that was our idea here. So putting the test to the theory, I will try to guide you to this. Oh, so this is with Kyrit. There's a small spelling error. I only see it now. Yeah. So here on the axis, you can see the entropy of the treatment. So that means the growth medium. Here we have the host. So that's the cyanobacteria, in this case, the plant structure model. And then we have the zoospores. And this is the particulate entropy. So that is what we measure with this elemental analyzer in the cell, or the total amount of the cell. And what we see pretty nicely, when we change the entropy of the medium, the cyanobacteria do the same. So that was a good start. But what we then see is with the zoospores for the parasite, we see that the changes are less evident. You can see here with a lower entropy, there seems to be no difference. And with high entropy, so with P limitation, we see actually that it is more plastic. So the host seems to be flexible to N and P, and the parasite seems to be mainly flexible to P and not to N. So this was first sort of confirmation, maybe indeed there's a certain limit that it can reduce its nitrogen and not lower than that. more chirates. So what we then see, if we then express, so this is the entropy of the host itself. This is maybe a very strange unit, but it is essentially the amount of spores produced per host, biovolume. Well, so with higher NP, there's more spores produced per volume of the host. So this also seems to match that trend. If the host is more nitrogen rich per volume unit, it produces more spores. But also what was very interesting, there was more spores, but they seem to be smaller. So we actually measured them, but we also measured their carbon contents. And then it seems to be that maybe there could be some trade-off with nitrogen, which is interesting. For yesterday, we had this discussion about how it's consuming the cell and how it may relate to the host density. So you can imagine this may have a lot of effect on that in the field, right? For finding the host. If you have bigger cells, maybe more energy, you can swim faster, or you have a longer lifespan, I don't know. Or with shorter, it's like luxury, right? High host density or high quality host. You don't need to look for a host long. I guess we will never know if it's really a decision that the chytrids make or whether it's just biology, but I think it will be very interesting to dive into this part deeper. Well, then another example of viruses. This is very complicated. It's from chemostat experiments. I'm not going to explain all of that, but what we essentially see is, if you go from the right to the left, that's basically this axis means P limitation. The black and the white dots is more CO2. So we see here there's some effect of adding more CO2, but the effect seems to be kind of dominated by the phosphorus gradient. Yes, so that brings us to more theoretical scenarios, because there's not so much data. Here we see again the carbon to nitrogen ratios. So we did some reviewing of all the work that we could find out there on autotrophic hosts. So the CN, if it goes down, means nitrogen addition, right? So more nitrogen goes down, and this means nutrient limitation. And this is the pathogen performance. So lower pathogen performance, no change, or higher. So what would you expect? In which spots would you expect the pathogens to be? So the fungi and the viruses. So if we add nutrients and the CN go down, where do you expect the pathogens to be? In which box? Which of the three? This one? So with more nitrogen, better pathogen performance. Okay, and the opposite, nutrient limitation. Hmm?
I'm seeing it before, so.
Well, you can also see it a little bit from the colors, right? So the green color was basically our hypothesis. So we indeed expect, if you add nutrients or nutrient limitation, that they should go along this axis from the lower left to the top right. We also did this for carbon to phosphorus, etc. But I will just show you nitrogen for today. So what we indeed see is for some fungi, we see they follow this trend here nicely on the upper right. You can see it's freshwater and marine and terrestrial. It's a bit of a mix. The opposite we could also find here with nutrient limitation. For some reason, there was more studies also there. And marine, maybe nitrogen limitation is more common in marine systems, I guess. So they will nice along this axis, but of course there's always some that don't follow the rule that we want them to follow. And more interestingly, we have this box here on the upper left, which is very interesting, right? That if you add nutrients, yes, if you add nutrients, sorry, I need to think sometimes myself with these ratios. If you add nutrients, that actually the pathogen may suffer, sort of, or that there's no response, which is a bit surprising, right? Could you think of a reason why this may be possible? I think we discussed it yesterday also a little bit. Defense. Okay. So yes, immunity. So or other cell processes that may counter the parasite. Exactly. So this is the picture where we stopped a few minutes ago. indeed, we can make this picture a little bit more complicating by more nutrients means positive for immunity. So the cell can defend itself, and then there may be a negative response to the parasite growth. But also the immunity may be negative for the host growth, because obviously it cannot use the nitrogen twice. So it's very costly, probably, right? Like enzymes, you need to invest a lot of nitrogen maybe in this immune response. There has been some work before also from Thomas Polak. He showed that with some knockout mutants, you can see that actually in the wild type, there's no problems which may be related to production of some biochemicals. And also some other work from Detmer and Karen here at NEO, showing that the response ratio for toxins, it's quite well known that with more nitrogen, relatively more of these toxins can produce or other metabolic compounds, because they also require a lot of nitrogen. So if there's a lot of nitrogen, maybe the cell has more money to spend, how to say it, it has more flexibility to also invest maybe in other things than just growth. Well, this brings me to the SI model. I was very happy that Alena introduced this yesterday. I don't know if you all remember, but it's a commonly used disease model. The big difference with this one is that it doesn't have the third box. What was the third box? Recovered. So why do you think we not use it here? Yes, it's a deadly disease, exactly. So we focus on the susceptible and the infected. Again, this beta is the transmission, and the Y was the recovery. There's no recovery. So what we did then, we discussed yesterday also a bit. It's quite a simple model in the basics, but by changing very small things, it gets very complicated very quickly. So what we do, are actually the people I showed you in the beginning from the C-Sync workshop. There's some real modelers in there, so to say. And what they did is changing this SI model with making the growth of the host dependent on this nutrient ratio. Also adding infectivity, which means with more nutrients, the parasite may be more infective. So like with making them more spores, essentially, or that spores are better at finding hosts if they are nutrient rich. And then the third one is the immunity, which means that maybe with more nutrients, the immune response of the host may be present or not. So these are the three scenarios that we will look at with this model. Well, it looks very complicated. I honestly also don't understand it. But the basics is that the host growth is dependent on nitrogen, and here you can see those transmission rates again. And it's kind of also a mass balance, right? The susceptibles get infected depending on this nutrient ratio or not. So we have four submodels. The first one is just basically the boring baseline model. So there's no effect of the stoichiometry. The second one is what I just said, infectivity. So remember that word: more nitrogen, more infective, more successful at infecting. Then the third one is immunity. So the susceptible hosts. So this box has a stronger immune response, because obviously when it's infected, it doesn't matter much anymore, maybe. And then the last one, a combination of both these models. So for this graph, we'll focus on the transmission. So with the model, we can look at a lot of stuff that's going on in the box, but now we will focus on the transmission. So this data, this is the infectivity submodel, and what you can see also nice in this graph. So this is the transmission. The blue means low transmission, the yellow high. So again, this is the nitrogen axis. So if you add more nitrogen here, you can see it gets a bit more yellow, which makes sense, right? Because that's how the model also is kind of formulated, right? So with more nitrogen, here you have the high NC ratios, there's more transmission. The K is the carrying capacity, but it's essentially the amount of carbon present. So at a low K, high N means a high NC. So we, in the models, formulated the opposite. So high NC means high nitrogen, which means also a lot of. Well, then the second submodel was with the immunity. So more nitrogen, more immune. You can see here the colors are obviously at the opposite in the top left. So with low NC ratios, or with generally a low nitrogen content, you can see the transmission is highest, which makes sense, right? Because the host can invest more in this immunity. What was nice to see is that you then see the reverse outcome. But then if you combine both, you get this kind of intermediate outcome. So if the host has immunity and if the pathogen also gets more infected with infectious with more nitrogen, we see that suddenly the transmission is here in the middle. So at the so basically this is also intermediate stoichiometry, right? So what I showed earlier from theory, this is kind of an intermediate stoichiometry, which also kind of matches this stoichiometric theory. Yes. Well, then putting the test to the theory, I went a bit fast actually. I'm almost through already, I think. But this is an experiment that Detmer did with some students and with the cultures from Silke. So then they tried to confirm basically what this model and this theory was showing. So we have a gradient of nitrogen availability. Obviously, then we will check the infections throughout time, which enables you to calculate growth rate, right, of the infection or of the host. Again, the idea is then that you will see this kind of optimum with nitrogen to carbon ratios with this infection rate, because on one hand there's immunity and on the other hand it's more infections. What I saw is that indeed with high NC there seems to be some kind of trend to lower infection rates, but if you look at the actual N quota, so in the cell, it seems to somehow match this trend. But yes, it's not, maybe Detmer want to comment on it later, but I think there's also still some work to be done here, but at least it shows that the trend is a little bit matching what we would expect. Well, I should have added more slides, huh? No, it's fine. So some of the take-home messages, I mean, stoichiometric patterns seem to be somehow predictable. They seem somehow to match a bit between fungiviruses or different kinds of autotrophic hosts, because it was also plants and algae. But the biggest problem probably still now is that there's like extremely limited amounts on parasite stoichiometry. It's based on a lot of assumptions. And I think with this Kitrid experiments that we did is one of the few. There's some theoretical models on the viruses, but also for the viruses, I guess there's still a lot of room for more measurements. Well, what's quite the strongest is that this reduced parasite performance under nutrient limitation. So the opposite with immunity, there's still also a lot of work to be done, which could be very interesting to do. But also again here with the fungi, there is a lack of data on phosphorus, so it's a bit more explored for nitrogen. But what is very interesting is that there seems to maybe be some kind of optimal at this intermediate nutrient supply. And I think this is very interesting because also in line with the paper that Alena showed yesterday, also with temperature or other environmental factors, there seem to be often some kind of optima, maybe because this parasite relies on the host, right? So I call it, there's like a certain window in which the pathogen can function if it's really dependent on that one host, if it's really parasitic. Well, that was it already. Or discussion points.
Yeah, thanks. Yeah, I wanted to talk a bit about the immunity and immunity mechanisms, because we talked a bit, just the two of us, but it's true that for cyanobacteria, there's, at least from what I know, there's not a lot of information about immunity mechanisms. There's some with diatoms and the programmed cell death when they get infected, but for cyanobacteria, I don't know if there's any data has been seen.
For immunity?
Yeah.
No, also not that I know. So we kind of relate to this toxin presence, but honestly, there's some papers that show, like from Thomas, I think that's from field strains, right, where he could show different. Yeah, they were isolated, and after that he showed that. But I think in the field he was observing also some kind of trends with toxin-producing presence and parasites, right? But there's also opposite papers already showing that even when there's a lot of microcysteines in this case, that sometimes there's still just infections. So I think maybe step one is there, maybe also first determining indeed what is this immunity actually, like which compounds are those. Yeah.
Maybe to add to that, I mean, Roland also looked more at the oligopeptides, the different parts of it, like not the typical toxins that you would look at, which might be then in this arms race, right, from the proteases, from the protease inhibitors from the host.
You mean that
Yeah,
then the parasite may have adapted already to digesting those?
Yeah, so then I think what he suggested is that there's like this ongoing arms race that parasite is investing into a new tweak on the protease so that it can actually digest through the, I always forget whether it's a cell wall or cell membrane. And then the host kind of makes a protease inhibitor to block that. But there's very little proof, and I think it's never been really taken up in many other systems, unless my mind is very much in Kitrid, so I'm missing the rest of the world. Yeah, I guess your plea for more data on anything, less reviews, more data.
Yeah, and it's plasticity, right? So not only the measurement, but also the, I call it the thresholds, right? Or like the how much flexible are they or not at all, maybe. Yeah. Serena?
Yeah, I don't want a bit more of complexity here, but just we were talking about also opportunistic behavior. So I don't know what is your idea about that, but I mean, a host that is too healthy can also be a bit too difficult. I mean, tricky for a parasite to attack. So I guess there is also a bit of that, but good balance, like immunity of the host, and also the ability of parasite to exploit some niche which are the hosts that are a bit weak and
Well, it always reminds me of the, is that with the Alexandrian system? I forgot what it was, but I mean, in nature, the community is so complex. I think there's probably always some of them that have immunity, right? Or some that have not. So then maybe the parasite, if it's a little bit flexible, it can still infect the ones that are not immune. But at the same time, if the community has enough, like with vaccinations, right, it can limit the infection enough that the host can still outgrow them, right? So
in the UK, they call that herd immunity. Yeah. It's quite trendy when we were all getting vaccinated with the COVID vaccines. Mm-hmm. And the other thing with immune response is that will have a cost as well. So the costs to fight back. And then there's a whole interesting dynamic then between this, cheats. You're going to get individuals that don't pay the cost of fighting back.
But so the community also functions as a whole to protect. So maybe there's some communication between the cells, right? I don't know. Yeah, I also know the Dynaflagy paper, but I forgot which one. That only some of the strains will.
Yeah, I think it was because of people who were in a certain community environment, and all the strains that this protected the susceptible hosts, because there were these other non-susceptible that were producing these chemicals that then provided them this chemical. So these are things that don't happen.
Yeah, we showed that for dinoflagellates and grazers in allelopathy. So if you have protist grazers that feed on dinoflagellates like Alexandrian, then the allelopathic ones can protect the non-allelopathic ones. It's related to associational defense, which you also see in plants that have protection against grazing, but then other plants can grow in between and are also protected. But then, there's also cheats that don't have the cost for these metabolites.
Also, just this stoichiometry, so to say, like we have this perfect microbial growth curve, right? But that would be so curious to see if you take a natural community, even if it's one species, how much variation is there on the cellular level, you know, in the content? Do you know what I mean? Like this axis of the CP, are they all very close? Are they all neutral limited at the same time, or is one of them limited and the other is not? You know, or with the lasers, huh?
Sorry, single cell. Yeah, you use this nano the relationship between chytrid infection and viral infection. If I play that out in my head, they are quite different in that when the chytrid attacks, it's assimilating the stock of biomass, but when the virus attacks, it gets the cell to make new viruses. So how does that interplay, or does that not matter? Or
yeah, good question. I guess it also depends on the virus, on the infection strategy, because I think there's some viruses that really hijack the host, right? But I think there's others that is more, how to say it? They use it as a growth medium, basically, right? Or like.
I think my interpretation of virus biology is the virus, by definition, must get the host to make new viruses, either RNA viruses or DNA viruses. So I can imagine having that a very different elemental stoichiometry story than a chytrid that just, you know, consumes.
Yep. And I think that's what makes this Planktotrix system maybe so interesting, because you can actually, what Serena showed with the fragmentation, I think the cells literally break where the rhizomes were growing of the fungus. You can see them growing inside, right? And that's where the cell die off, but the rest still remains alive. So in that case, also the discussion of neutral limitation with the chytrid, I mean, it can adapt by just growing further into the filament, right? But maybe some other systems like the Astronella, where the cell is limited, it cannot, how you call it, it cannot decide to from one infection to grow to the neighboring cells, right? So it's maybe this neutral limitation effect may also be different between these host systems.
And maybe some other tweak to that, because about how infections actually change, maybe I really love that, but also like how it changes actually the activity of the host itself. So even in the infection, it might change the nutrients uptake or the and I could show you some things, but we kind of recently observed something interesting with the cyanobacteria system. So it's a Dolichospermum host, and when the parasite infects, and you see with all the fluorescence, so with the particle singing, when the infection starts, the cell that gets infected, it really it's much more bright. And then you really see how the rhizoid is going through, and that part is getting bright. And then, of course, when the parasites kind of develops and gets mature, then the cells die at some point. But you really see how this rhizoid is progressing, and then always like makes the cells. And at this point, and this is maybe something interesting in physiology, because in the beginning I thought that, oh, these cells are maybe doing more photosynthesis, so maybe the infection is actually kind of increasing activity. But I'm still not kind of unsure, but it can also, of course, be some energy like heat, like dissipation that creates this. So I don't know if somebody knows like how.
But yesterday, Mike, you showed something with the fluorescence, and we observed it also with the phytofarm. I mean, there is a time where the cells is still active, and you see it that there is not an immediate response in the photosynthetic activity.
So I think the point is that you get an increase in activity, right? That's what
yeah, it's really the infection on a single cell, so you see this the cell that gets infected changes.
No, sure, but what the means that there is not an immediate decline. So there is probably a response also of the cell to the infection.
Then it relates to Michael's question. Maybe does the chytrid hijack the host cell and then force it to increase photosynthesis? Or no? Yeah.
Measure its activity on this single cell. So I think there are some ways to do that.
But I think that maybe they consider hybrid as a single entity that does not have different strategies, like R or K strategies, that maybe it can be that some species flourish when there is rich nutrients and like increase photosynthesis to just survive. And then other, I have like a higher sporulation rate where the nutrients are down. So there are strategies and key strategies also on a community level, because now we're thinking about just one strategy and one response, but if you analyze communities, then this might change.
So something that's observed with the temperatures. Do you see difference in spore size and also spore size number with higher or low?
As I was thinking from the ascomycetes, where like you have really high rate of reproductive spores, but the nutrients are low because you have arrived at the end of the availability. So your only way is to try to be the most effective, like use everything for reproduction. But when the nutrients are high, then just grow vegetative. I don't know much about the parasites. Those are the parasites, but maybe you don't have this kind of shifts between life state and life modes. So can they really have it to stay in the cell and flourish? And
there is a limitation. They do have a development time. Was it with temperature?
If they use the photosynthetic apparatus, do they use them not just for reproduction, but just to grow inside the cell?
And sometimes they grow, but they reproduce mechanisms. But it's not that, of course, the size can change, but and the size of the sporania, which might be proxy also for the amount, can change. So there might be some kind of flexibility of maybe more and more within, but I think at some point,
Just maybe.
Yeah, it's a good question. I don't know what then triggers the maturation. Like at some point I don't know how fixed it is, how many
but you don't see a lot of variability, right? It's not like at a certain temperature with certain nutrient conditions you would see anything from 1 day to 5 weeks or something like that. It always stays quite in the same time window. And also, if you inoculate nicely, I think that's what you showed really nicely as well. If you inoculate with zoospores nicely, you also kind of have very synchronized zoospore release events. There doesn't really seem to be a lot of individual choice in the Gatrid, at least, of the phytoplankton. Or maybe other groups.
There's an online question from Andre. I don't know.
Can you hear me?
Yes.
Thanks, Thijs, for the presentation. Of course, I'm happy to see a model there, but it also is very risky to show a model when I'm listening. So
yeah, I know.
I had a question about it, and can you show the equations that you just showed?
Now it would be easy to say, "Oh, we are losing connection."
Oh. Yeah, I know. You can say you can say that.
Can you see it?
Yeah, I can see this, and I was
These
more.
ones?
So show me the 31. Well, it's both these things actually, because one of the things that is here.
Oh, this one. Sorry.
No. It's the 31 or 30 and 31. So what you do here is, of course, you take a very often used model for the growth of cells, but now you actually add an equation for that infected cells also contribute to population growth rate. And isn't this at odds with like what these Gitrids actually do? Because you have these Gitrids infect a cell. They cannot divide anymore, these cells. They cannot really give rise to new cells. So I'm curious about like why you add this growth from I term there, and it's also it's something that you don't show here, but I just looked up your paper, Detmer, that there is something strange about going on about the quota that you actually the nutrient quota in the cells that you assume, because when I look at the model, I think there are two questions. First of all, do infected cells actually contribute still to population growth? Do they divide? And how do you envision this? And second of all, in your paper, you actually then assume that, you know, the growth from these infected cells turns into susceptible cells with the quota of the susceptible cells. So somehow the infected cells somehow change their nutrient quota at the moment that they contribute to population growth. Both assumptions I find actually not very well aligned with what the empirical situation is.
Yeah, no, that's true. So for chytrids, so the model is developed really for a general primary producer disease model. So that's where we came from, and there, yeah, we just assumed that in some case, well, there is also from rest of plant literature proof that infected cells still grow as they still continue growth. So it's not per se deadly to the whole infections, to the whole host cells or host organs. So we took it from there, and then we just and characterized it with the chytrid. So you're right that you could argue that a cell by itself is infected, but then you can also think about a filament that is not. So then I wonder how that, but either way, maybe it's then you should place it in early infections or so. So if a cell then still divides, with any cell division with phytoplankton, you get a redistribution of your elements. So this shift towards susceptible, I think that is okay. I think the only point you're right in is that if the infection is really into the cell, so if it really continued, so it's not just like starting infection, but really entered the cell, then I guess that's a wrong assumption, right? So you should be careful with that. Yeah. And then you get
it's one of my problems always with phrasing models is that there is too easily you just take a model from the literature and you tweak it here and there a bit without really thinking about like what's going on in the biology actually of the problem, and it's quite hard to rigorously translate biology to an equation, and it's an underappreciated aspect, I would say, of modeling. So yeah, I see your point that this is what's being done.
That's not what I said, Andre. It's not what. It's just what we developed with the CSync group. We use this model also in systems. So I think the issue is that we just made it so general that then, of course, you have to make it specific again if you zoom in specific more.
If you zoom in, okay.
I think that's where so we developed this model really by ourselves, and we made these decisions explicitly for this, right? Based also on grass and virus diseases, just in general to have a disease, nutrient-mediated disease model, really.
Okay, yeah.
Applications, including the chytrids, and then we could have said, okay, we actually take out this component for the infected, right? And then you have a more rapid mortality, etc. So yeah, you could have done that.
Yeah. Okay, in that way. Okay,
but technically, we also wanted to show with this model, so we parameterize it for the chytrids, but we also wanted to show that this idea still is that it's a general model. So in the paper, we also don't show the examples of like the chytrid system later. We don't compare it with the chytrid system by itself. We just try to keep it general in that aspect. But yes, to mention that if you want to use this model, you should maybe take out that component and make the infected directly death, so to say. Yeah.
Okay, thanks.
So then I also did just about the comment with the viruses and the chytrids. I think that's an interesting point also with the system and the way we looked at it is that the viral particles that they're basically nucleic acids, right, with a protein capsid, so to say. So that's what Thijs mentioned about like high demands for nutrients and especially phosphorus. So I think it's interesting to see that so basically a chytrid in that aspect becomes more like a grazer. It just has to deal with what is available, right? It just destroys it and takes it up. Whereas a virus is depending on the host machinery and what is available for the host to assimilate into viral particles, right? And I think and then if you take that alone and you compare what a host would need in terms of nutrients and the virus, then the assumption still is that the virus will be more affected relatively, so it will produce less new viral particles compared to what a host would produce as new cells relative. That would be kind of and of course not the number of particles, that will be way more anyway, right? And viruses can still progress massively, there will still be a lot of viral particles, but the relative impact may be bigger, so to say. That would be one of the assumptions there.
And in the model systems that you're planning to use, can any of these algae do phosphorus remodeling from lipids? There's some algae, the phosphorus starved, basically recycle their own P-rich lipids to make S-rich lipids, so that they don't have a P demand, but they can recycle their own P. Can they do that?
I don't know, and that's a good point. So I would expect so. So with we only have some for microcystis. We have some circumstantial evidence that we see the shift with P limitation. We see a very strong increase in sulfur in the cells, and we still want to analyze that on sulfolipids and phospholipids to see if there was a shift from phospholipids to sulfolipids, because that's one of the mechanisms how to deal with phosphorus limitation, right? So that would be one of the things to test for those. And there might be alkaline phosphatases and some else as well to actually get organics. So if there's stock dying, they can still, and there's organics, they may still be able to take that on.
Do you measure the total phosphorus, or is it, phosphate or whatever that?
In this?
In the planned experiments, is it total P or it's total P?
Well, in planned experiments, we did not explicitly plan things here, and all the CP ratios, it's all particulate. Yeah. But for the planned experiment, we still have to see, and Thijs tried with a colleague to get actually with the Plantotrix chytrid system, actually to also get a virus on it. So we tried on the field to get a virus infections, and then what I think is interesting is both the competition between the two for N and B and test hypothesis that Thijs showed, but also if you have infected virus infections, then the chytrids can of course also reduce maybe the viral load by actually infecting virus infected cells, and then they will also destroy the viruses, right? So anyway, but that gets way more complex. And for the photosynthesis and cell size things, I wonder how many of these things are like a change in photosynthesis with chytrid infections and maybe in general. So light is often we consider light as something that phytoplankton needs, but at the same time it's also actually often a stressor. That's why we see all these additional pigments being involved. Then we think, oh, they look healthy, but actually they are light stressed, and they need these extra pigments to reduce that light stress. Of course, at the same time, sometimes they're light limited, so they need to have these pigments. And I can imagine that if you take out machinery for the light energy to proceed, so to say, all the reductants and so to actually use that energy, you get a blockage of the whole photosystem. And light just doesn't stop, and they cannot block it. If they still have pigments, the light will keep coming in. So then fluorescence is one of the photostress signals, right? So it's photooxygenic stress release, so to say. So that would be one of the coping mechanisms.
Yes.
And the same for size, I wonder. So we also see often that things get bigger when growth is arrested, and not because they just want to get bigger or it's per se a strategy. It's just that something limits growth, but the rest is still continuing. So if they have storage possibilities, it will become bigger, just because of that process. Growth is like arresting, but they still accumulate things. And if chytrids can do that, you can get changes in cell size too, or spore size. So that's just some, like, which parts are real strategies and specific traits of certain species or genotypes within the chytrids, and which aspect is like just general kind of a result of this like arrested growth and then accumulation of lipids or whatever. I don't know, that's just some thoughts on those things. And then I think, yeah, it's time. So,
But just to add on to that, so you're actually linking now the lipids to these castrating parasites. Because that's basically what that.
yeah, that's what, yeah, so in a way, that's what the zombies also, it's I think what you mentioned yesterday too, right? It's in a way, if they can use photosynthesis, of course. But it might also, yeah, I'm not sure if that really happens. But anyway, let's conclude this part. Also put a line